Plants, like all living things, need to reproduce to perpetuate their species to survive through time. So today we are going to get to know plant asexual reproductive strategies. Artificial methods of asexual reproduction are frequently employed to give rise to the new and sometimes novel plants. They include grafting, cutting, layering, and micropropagation.
Another example of using artificial method is through plant tissue culture. So by doing plant tissue culture, you can use it on most plants. So what do we need? What type of plants or what part of plants that we need for plant tissue culture? You can use the leaves, the roots, and even the stems of a plant. So the first thing that we have to understand is when doing plant tissue culture is you can either bury it in a soilless medium, which is very rich in nutrients, or you could put it in a sterile condition where you have to fulfill three things, which is it must be full of light, humidity, and temperature. So during those time intervals when you're doing the plant tissue culture, you have to continuously subculture the plants, the plantlets, so that it will be easy to propagate them.
Now, we are going to the advantages and disadvantages of asexual reproduction. For asexual reproduction, the advantages is first, it will help to increase the population rapidly. Number 2, the energy efficiency. Number 3, the cost is effective. And number 4, the time taken to be propagated will be decreases. And the last one for the advantages is no need to plant to be propagated. So when the plants are under stable condition, which is lab conditions, it will help to increase the growth and the productions.
One of the major disadvantages of asexual reproduction techniques is that they also propagate pathogens. Pathogens can be viruses, bacteria, and fungi. How do they work?
Talking about the disadvantages, there's no variation or diversity in the population. Because we are using the method, simply can say as a cloning method, so it is identical to the parent plants, so there's no variation in the plants. And if let's say if there's any variation, it can be positive or negative mutation. If negative mutation means it can the whole set of the plant will lead to a bad effect.
How do we maintain plants to be disease-free? So first, we have to select a mother plant. So for the mother plant, we have to make sure that it's disease-free, very high in quality, and also it also needs to be a younger plant.
After we select the mother plants that fulfill with our criteria, we will clean and seal them under lab conditions. There are several steps that can help to maintain the plants disease-free. So, first, which is we will use chemical. The chemical that usually we use is oxides.
The second step is quality check by the expert daily. This can ensure that there is no fungus and microorganisms that live in the plants. If there is microorganism, it will be eliminated at first.
The last step is properly clean up the contaminated plants. So, these three steps can help to maintain the plants free disease.
plants are right, but not exactly like their parents. So, Miss Camille, how is plant vary from their parents?
Okay. So to propagate the plant, we will regularly subculture. Means we take from the plant, the mother stalk. So we will regularly subculture to more plantlets, to get more plantlets. This will be done at the month interval, or simply say 4 weeks intervals, and also choose quality stock for the next propagation.
Another way that we can produce more variation on plantlets is we use more chemical radiation, such as cocaine. When we use more chemical radiation, we will produce more hormones. So when more hormones are produced, it leads to more mutation. Also, we can also increase our very own subculture. So when we increase our subculture, we can see the difference in plantlets from the mother plant.
Since tissue culture is an important technique for propagating, so there are some examples of fruits or vegetables that applying tissue culture to propagate. First, banana. Banana, which is a sterile hybrid that cannot produce seed, is important for the fruit to be propagated by using tissue culture. And the second one is aquatic plantlets. As the conclusion, many botanical gardens and universities use tissue culture for plant conservation. The Atalanta Botanical Garden has a tissue culture lab that propagates native species of orchids and lilies. Tissue culture of rare species is also used to help increase the population in the wild, as they are replanted in native habitats. So, our title is answered: yes, new plants can form without the production of seeds and spores.
That's all from us. Thank you.